बाद में किसका है?
हाँ?
उसके बाद किसका है?
दोपहर दोपहर को
दोपहर को कौन है? 30 ये बाहर ले आओ.
हाँ सर, you can put it on the desktop.
कौन? No. Good morning. Do you all hear me?
Yes, sir, you are audible.
Okay, thank you. Morning. I'm starting. Today's lecture is nervous tissue. In this lecture, we'll cover about the neurons, nerves, and the neuroglial cells. So, first of all, we are familiar with nervous system. It coordinates the various activities of the internal organs, and it also keeps us informed about our external environment, changing external environment, and prepares us to cope up with the adverse situations in the external environment. So anatomically, this nervous system is divided into a central division and peripheral division. Central nervous system comprises the brain and the spinal cord, whereas the peripheral nervous system comprises the cranial nerves, spinal nerves, and the peripheral nerves. Functionally, the nervous system can be divided into somatic nervous system and autonomic nervous system. Somatic nervous system means it provides sensory and motor information to all parts of the body except visceral system, the smooth muscles, cardiac muscles, and the glands, which are supplied by the autonomic nervous system. Somatic nervous system is entirely voluntary, within our control at will, whereas the autonomic nervous system is involuntary. We cannot exercise any control. This autonomic nervous system is again subdivided into sympathetic, parasympathetic, and enteric nervous system, and we'll see little bit about this. Now, composition of the nervous tissue. Nervous tissue is composed of neurons and the supporting cells. The supporting cells in the CNS are called central neuroglia, which are astrocytes, oligodendrocytes, microglia, and ependymal cells. In the peripheral nervous system, the supporting cells are called peripheral neuroglia, like Schwann cells, satellite cells, and enteric neuroglia. Besides, there are blood vessels to supply the nutrients to the neurons and glia, and connective tissue elements that support the neuronal structures. In the nervous system, there is a blood-brain barrier. That means a kind of barrier that prevents exclusion of certain materials to enter the blood system and their escapes into the neuronal parenchyma. So this blood-brain barrier will be dealt a little bit, how they are formed. Now, before we describe anything into detail, two terminologies are important. One is called afferent neurons and their fibers, or simply afferents. This means the sensory neurons that conduct impulses from the periphery to the central nervous system. So these are afferent fibers or afferent neurons. Efferent neurons, on the other hand, are the motor neurons that carry information from the CNS to the effector cells towards the periphery. So these two are to be remembered, afferent and efferent projections. Now, based on functions, neurons can be sensory neurons, motor neurons, and interneurons. Sensory neurons, under this there are two types: somatic afferent neurons, whose fibers carry sensation from the body surface with relation to pain, touch, temperature, and pressure, and also from proprioceptors, which are present in the muscle spindle, joints, and tendons. These proprioceptors provide sensation of the body position and the muscle tone. The visceral afferent fibers carry sensory informations from the internal organs, mucous membrane, glands and blood vessels. So this information goes to the brain. Motor neurons on the other hand carries information via somatic efferents to the skeletal muscle, while the visceral efferents carry motor impulses to smooth muscles, cardiac conducting cells and the glands. With this there can be contraction of smooth muscles, control of cardiac muscles and release of secretion from the glands when the visceral efferents work. The third type is interneurons that integrate information from sensory to motor neurons. Now when these three work together, there constitutes a what we call neural pathway, that is the reception of stimuli by the sensory afferent fibers, their interpretation in the CNS and the initiation of responses by motor efferent fibers to cause an effect like constriction of muscles. So this constitutes what we call a neural pathway. So this is one diagram of a neuron. This is in fact a motor neuron. You are familiar with the parts, like this is the cell body which ejects several dendritic processes and then one long axon. Axon may be myelinated or unmyelinated. The term cell body means the part of the neuron that contains the nucleus along with the surrounding cytoplasm. Dendrites are peculiar in the sense that as they emerge from the cell body, they gradually taper. So this is one of the identifying feature for the dendrites. So they will gradually taper. On the other hand, axons are singular structures and their diameter is constant throughout. Now how impulse is conducted? Impulses are collected from the peripheral organs and then they reach to the dendrites. From dendrites they go to the cell body and from there information is sent to the effector cell, like here skeletal muscles, via axons and synaptics terminals which are present on the effector cells, like here skeletal muscle. Based on the morphology, there are unipolar neurons, bipolar neurons and multipolar neurons, and we will see how they are classified and how they look like. So first of all, unipolar neurons. Unipolar neurons have a singular process. Although you can see this process is divided, this is considered as a singular process, in fact an axon. So unipolar neuron, which are sensory neurons, have just one axon that is divided into a long peripheral process and a short central process. Now the peripheral process contains peripheral arborizations that act as a receptor, means that collect information from the peripheral sites like skin, muscles, tendons and joints, and this information travels via the peripheral process to the cell body, which is located inside the dorsal root ganglia. This dorsal root ganglia is located in the dorsal root of the spinal nerve, and from here the process that projects via central process to the spinal cord and terminating in the dorsal horn, which is sensory in nature. So all sensory neurons are in fact unipolar, and they are located in sensory ganglia like the dorsal root ganglia and the cranial nerve ganglia, which are associated with the cranial nerves 5th, 7, 8, 9 and 10. So these are all sensory ganglia and they contain unipolar neurons that carry information from the periphery and reaches to the spinal cord. Bipolar neurons, on the other hand, are involved with sensation of smell, sight, taste, balance and motor coordination. So they are present in the olfactory epithelium, in the retina, in the cochlea, the spiral ganglion of the vestibulocochlear nerve, 8th nerve, and in the cerebral cortex involved with motor discharge. So this is one bipolar neuron having an axonal projection and a dendritic projection. This is the cell body. Multipolar neurons, on the other hand, are having multiple branches that emerge from their cell body. So we can see processes that can be categorized as dendrites. So these are called apical dendrites, and then also there are basal dendrites present in the pyramidal neurons, not in all multipolar neurons but especially in the pyramidal neuron, and there is one singular projection, that is the axon. So this is one pyramidal neuron of the cortex, and this can be seen, the whole profile can be seen when we treat the tissue with silver nitrate solution called Golgi stain. So this motor cortex are these motor neurons, multipolar neurons are essentially motor neurons, and they are present in the motor cortex like the pyramidal neurons are located within the motor cortex. Another example of multipolar neurons is Purkinje cells. These are the Purkinje cells. And the third example is the ventral horn cells or anterior horn cells within the spinal cord. This is one cross section of a spinal cord. Its grey matter is divided into dorsal horn, lateral horn and ventral horn, and in the ventral horn you will see multipolar neurons. They have many processes. So these are examples of multipolar neurons: pyramidal neurons, Purkinje neurons and the ventral horn neurons of the spinal cord. Structure of neurons. You are familiar little bit with the structure of neurons. The information is essentially same, but you need to know little bit extra at this stage. Like the neurons are characterized by having a large oval nucleus, and this nucleus is always euchromatic, that is highly transcriptionally highly active, and there are prominent nucleolus. Here you can see, and you can see a number of organelles. Like the cytoplasm will contain Nissl bodies. Nissl bodies are stacks of rough endoplasmic reticulum and the ribosomes. So this can be stained with a basic dye. So these Nissl substances are basophilic in nature. So these are the Nissl substances that are basophilic. So these are actually stacks of RER and the free ribosomes present in the cytoplasm. Neurons are rich in cytoskeleton like microtubules, intermediate filaments and actin. Now you have to know the distribution. Microtubules are mainly present in the dendrites, whereas intermediate filaments in the form of neurofilaments are present in the axons. Actins are present in the cell body and as well as in the dendrites. The dendrites contain rough endoplasmic reticulum but no Golgi complex. On the other hand, axons contain smooth endoplasmic reticulum. So these are some information that you need to know about the cell organelles in the neurons. In the neuron there is one structure called axon hillock that contains no organelles, but at any stage you can see some cytoskeletal elements to be present in axon hillock temporarily. They are en route towards the axon, so they are not the permanent structure in the axon hillock. I will come to that axon hillock. Now, dendrites. Dendrites are always multiples and forming dendritic branches, and in some neurons they form what we call dendritic tree, like in the cerebellum. The Purkinje neurons form extensive dendritic branches forming dendritic tree. These dendrites may contain spines or some outlets from these dendritic structures that I will show. This is one structure that can be seen after Golgi staining, as initially done by Camillo Golgi. That's the portfolio of Camillo Golgi. Axons, on the other hand, are single and originates from a particular region called axon hillock. This is the axon hillock where the cell body terminates and the axon begins. So that particular segment is called axon hillock, and this is essentially devoid of any organelles. Then this axon, which originates from the axon hillock, can be smaller or even longer, much longer, and based on this length neurons can be Golgi type 1 whose axons are much longer, and type 2 whose axons are very small and restricted within a localized structure. There is one segment called initial segment. This region of the axon where no myelination happens is the site where the impulses are first generated. Initial segment. So this is the region where impulses are first generated. Because this region of the axon, this region contains clusters of voltage gated sodium channels. So depolarization wave will be set in, and ultimately this will be conducted along the axons. Based on myelin presence and absence, there may be myelinated and unmyelinated axons. So these are axons with varicosities, and these varicosities are nothing but the accumulation of certain organelles during their transport called axonal transport. We will come to that point. So I believe by now you can distinguish the dendrites from the axons from these features, light microscopy, and also from little bit of electron microscopy, and also from physiology, like dendrites they can generate impulses, but axon they can conduct impulses. So these are the differences between dendrites and axons. Now, as I told you regarding the transportation system in the neurons, neurons synthesize lots of proteins and other peptides, etc., and so they need to be transported to different regions of the neuron. So this is done by a transport system. Traditionally this is called axonal transport system, although the transport material also reaches to the dendrites. So this is called axonal transport system, and it has two systems, anterograde and retrograde transport system. Anterograde transport means from cell body towards the axon, and retrograde transport is just the reverse, from the axon towards the cell body and from dendrites towards the cell body. So anterograde and retrograde transport system, this transport system is mediated by some microtubule motor proteins like kinesin and dynein. Kinesin is a microtubule motor protein involved in anterograde transport, that is helped in transport of materials, and dynein is a microtubule motor protein involved in retrograde transport. Based on the speed of the material transported, there can be slow axonal transport, in which the rate is about 4 millimeter per day along the axon, or it can be fast axonal transport, where the speed is 100 times more than the slow transport, 400 millimeter per day. This slow axonal transport is always anterograde, that is from the cell body towards the axon, and mainly transport material includes the cytoskeletal proteins and also intact cytoskeleton like actin, microtubules, etc. On the other hand, fast transport is bidirectional. It has one anterograde system and it has a retrograde system. By fast anterograde system, the materials which are membrane bound like mitochondria, synaptic vesicles and smooth endoplasmic reticulum are transported to the axons. On the other hand, in the retrograde system, many materials are retrogradely transported towards the cell body, and this could be the endocytosed vesicles after synaptic neurotransmission, the proteins, the leftover proteins, and also some neurotransmitters which are transported back to the cell body by first retrograde transport system. This is also the mechanism, fast retrograde transport system, by which the viruses and toxins use and enter the central nervous system. So you remember this thing. Synapses. Neurons communicate with each other via synapses and also with their effector cells like mast cells and glandular epithelium, which are the effector cells, via the synapses. Synapses can be studied by electron microscopy and special technique like Golgi. With Golgi, we can see this kind of synapses, the terminals, axon terminals, which are present on the cell body. This is one neuronal perikaryon or cell body having these axon terminals. So synapses are formed on the cell body. So this type of synapses are called axosomatic synapses, axon to synapse. There can be axodendritic synapses, like here these are the dendritic branches, and axodendritic synapses can be seen here, dendrites and axons, axodendritic branches. This is axosomatic synapse, axodendritic synapse. There can be axoaxonic synapse and dendrodendritic synapses. So these are the 4 types of synapses seen in the nervous system. And regarding types of synapses, there can be chemical synapses, synapses that utilize neurotransmitters for communication between the neurons, and there can be electrical synapses like gap junctions, which are present in smooth muscle cells and cardiac muscles. These electrical synapses or gap junctions are made up of some channels which are called connections, gap junctional channels which are called connections, made up of some connexin proteins, hexameric connexin proteins. So as they open, they can permeate small molecules like glucose, ATP, and some ions, and also electrical transfer across this junction is possible. So this junction is about 2 to 4 nanometer, what we call gap junction. So this is another type of synapse where neurotransmitter is obviously not involved. Neurotransmitters are involved in chemical synapses. So at this stage you need to know little bit about the synaptic ultrastructure. Details will be taught in physiology and biochemistry classes, but you read little bit at this stage. So what are the components of a synapse? A synapse is composed of a presynaptic terminal and one postsynaptic terminal. So here presynaptic terminal, suppose this is one axon and the postsynaptic terminal is one dendrite. The presynaptic terminal can be identified by the presence of synaptic vesicles. So these are the synaptic vesicles loaded with synaptic neurotransmitters, and in the presynaptic terminals you will see presynaptic density where some specialized proteins are located and they form what we call active zones. Active zones are the areas in the presynaptic terminals where only the vesicles can dock and fuse. See here the vesicles fuse and these vesicles are coated with some protein called NSF that can bind to here the receptors present here for this NSF. Then the vesicular fusion occurs releasing the neurotransmitters at the synaptic cleft, and these neurotransmitters then bind to the receptors present on the postsynaptic element like here dendrite. So these receptor elements are clustered in the form of what we call postsynaptic density. So this membrane would appear denser when the receptors are clustered here. So this picture show about the axon terminals and dendrites. So one kind of synapse, the presynaptic terminals called clear vesicles, containing clear vesicles like glutamate, containing clear vesicles. At the same time we can have dense core vesicles that contain neuropeptides and substance P. So these are dense core vesicles, and in certain regions you can find ribbon synapses like in the retinal photoreceptor cells and in the hair cells. You will find this kind of structures, presynaptic structures. They help in docking of the synaptic vesicles to the structures and their fusion to the active zones. Now, based on the transmitter materials and proteins in the presynaptic and postsynaptic units, there can be asymmetric type of synapses and symmetric type of synapses. So what do they are? In asymmetric synapses, which are called type 1 synapses or excitatory synapses or Gray 1 synapses, as first described by Edward Gray, here you will see that postsynaptic elements is dense than the presynaptic density. So this is called type 1 synapse, which are excitatory, and they mediate glutamatergic and acetylcholine transmission. Like here you can see this is the presynaptic axon terminal and the postsynaptic dendrite. This dendrite is in the form of a spine, and here the postsynaptic density is more than that coating in the presynaptic ending. So this is one asymmetric synapse, type 1 synapse or excitatory synapse. On the other hand, there is one called symmetric synapse in which the density of the protein density in both presynaptic and postsynaptic membrane is almost similar, so they look symmetrical in protein distribution. So that is called type 2 synapse, and these are inhibitory synapse, like they utilize GABA in neurotransmission, GABA and glycine in neurotransmission. So inhibitory synapses are symmetric synapses. Here you can see that in asymmetric synapses that bear clear vesicles, these vesicles are round, perfectly round, spherical. Whereas in the symmetric synapse, which are inhibitory synapses, the vesicles are not perfectly spherical. They are somewhat quadrangular, as seen with the GABA or glycine vesicles, synaptic vesicles. So this information is to be remembered. Now neurotransmission happens via vesicular exocytosis, that is known to you. This synaptic vesicle fuse with the presynaptic membrane and releasing the transmitter molecules to the synaptic cleft. So this is one method by which ample neurotransmitter can be released. There is no control. But recently one type of exocytosis variety has been described. This is called porocytosis, in which the neurotransmitter release can be controlled or regulated via the formation of a pore. So that is why it is called porocytosis. That is, there is a formation of pore by the membranes of this synaptic vesicle. The entire thing is synaptic vesicle that fuses near the presynaptic active zone. This is the presynaptic plasma membrane, and it is firmly anchored by snare protein complex, V snare attached to the vesicle and T snare attached to the target presynaptic membrane. So these vesicles will tether, bind, and then they will reorganize with the presynaptic membrane in a way that a pore of 1 nanometer is created, through which these synaptic materials are discharged in a very small quanta. So this is a regulated neurotransmission recently discovered in the nervous system. Now there are various neurotransmitters, and many of these are familiar to you. What do these neurotransmitters do? So first, these neurotransmitters open the ligand gated or neurotransmitter gated ion channels at the postsynaptic membranes. Once these ligand gated ion channels are opened, then there is a conformational changes in these channels, and this will permit entry of ions like sodium ions to the postsynaptic membrane. That is the broad function of neurotransmitters. So there are excitatory and inhibitory synapses. Under excitatory you can have acetylcholine, glutamate, serotonin, inhibitory GABA and glycine. Neuromodulators are there like substance P, VIP, polycystokinin, and nitric oxide is one molecule that is not packaged within the synaptic vesicles and released immediately after its synthesis. In the excitatory synapses where excitatory neurotransmitters like acetylcholine or glutamate or serotonin is released, this excitatory synapses leads to generation of what we call excitatory postsynaptic potential on the postsynaptic unit, because suppose glutamate binds to the glutamate receptors at the postsynaptic elements, that leads to opening of the sodium channels at the postsynaptic membrane, leading to local depolarization and continuing the propagation as excitatory postsynaptic potential, action potential. At the same time, if in the inhibitory synapses, if some inhibitory neurotransmitters are utilized like GABA, this will lead to opening up chloride channels, and once chloride enter, that leads to hyperpolarization of the postsynaptic membrane physiologically, which is called leading to the generation of inhibitory postsynaptic potential, in which synaptic transmission slows down. this neurotransmission can be blocked, or there is a blockade of neurotransmitters even in natural mechanisms like botulinum toxin, which is derived from one gram-positive bacteria, Clostridium botulinum. This inhibits acetylcholine release. Not now, please. Okay, you can ask via WhatsApp. So botulinum toxin inhibits acetylcholine release. So this leads to muscle paralysis because this acetylcholine cannot bind to its receptor, nicotinic acetylcholine receptors present in the neuromuscular junction. So this will inhibit. Botulinum toxin will inhibit neurotransmission. Now in myasthenia gravis, which is an autoimmune disease, there are antibodies against acetylcholine receptors. So acetylcholine receptor is present on the postsynaptic membrane, and there are antibodies against these acetylcholine receptors that bind to this receptor site. So acetylcholine cannot bind to those receptors. So this leads to muscle paralysis, as happens in myasthenia gravis, a dreadful disease. Now after neurotransmission is over, the left out neurotransmitters are taken up by a mechanism what we call high affinity reuptake mechanisms. Okay, so that duration can be postponed, duration can be minimized. Otherwise, if this receptor neurotransmitter molecules are not uptaken, there will be continual discharge of impulses to the postsynaptic terminals. So the neurotransmitter molecules are reuptaken by certain sodium dependent transporter proteins present on the presynaptic terminals. Now after their reuptake by these high affinity mechanisms, the neurotransmitters like catecholamines, dopamine, adrenaline, noradrenaline, all those substances are again packaged in the synaptic vesicles, in the presynaptic axon terminals. The excess is then destroyed by catechol orthomethyl transferase or monoamine oxidase present on the mitochondrial outer membrane. So these will inactivate the left catecholamines. Now there are certain pharmacological agents like cocaine and amphetamine that interfere with catecholamine reuptake. So what will happen? These substances, these neurotransmitters will be there in the synaptic cleft, and they will again activate and stimulate the postsynaptic receptors. So there will be continuous firing of the postsynaptic receptors. Essentially is the method and mechanism that the drug addicts utilize, so that there is a continual stimulation of certain brain areas. So these cocaine addicts and drug addicts persons can know better how they feel after having these drugs. So they actually interfere with catecholamine reuptake. Acetylcholinesterase is one enzyme synthesized by the postsynaptic unit, and then it is released in the synaptic cleft, where it will degrade acetylcholine into acetic acid and choline, and choline is then reuptaken to the presynaptic terminal and packaged into synaptic vesicles, and these neurons are called cholinergic neurons that utilize acetylcholine. Now there are certain disorders where neurotransmitters may be lacking, like acetylcholine may be lost from the hippocampus and entorhinal cortex in Alzheimer's disease, and dopamine may be lost, or there may be decreased synthesis of dopamine in the striatum, leading to what we call Parkinson's disease. Now a little bit about the peripheral nervous system axon. Myelination is mediated by Schwann cells in the peripheral nervous system, and in the central nervous system it is mediated by oligodendrocytes. So what happens in myelination? The axon first comes to lie in a groove in the surface of the Schwann cell. This is one Schwann cell. Schwann cell. So as the axon is covered by the Schwann cell membrane, there is membrane polarization called abaxonal domain and adaxonal domain. Abaxonal domain is towards the extracellular aspect and adaxonal domain is towards the axon. So as the ab and adaxonal domains fuse, they form the mesaxon. This mesaxon then continues to grow and spiral in a anti-clockwise fashion until the entire myelin ring is formed. As the myelin sheath is complete, then there is formation of what we call outer mesaxon that connects with the narrow extracellular channel. The inner mesaxon is still there, that is in continuity with the adaxonal membrane. Now during this myelin formation, cytoplasm is left out in certain pockets of the myelin, and as the cytoplasm is extruded, the inner leaflets of the plasma membrane, as the cytoplasm is extruded, the inner leaflets of the plasma membrane fuse, forming what we call major dense line, these black lines in the myelin. Now these black lines alternate with the slightly less dense lines, which are called intra-period lines. Intra-period lines, which are formed by the fusion of the extracellular aspects of the plasma membrane. So ultimately these major dense lines and intra-period lines are formed in the myelin sheath. Now there are proteins like P0 proteins, myelin basic protein, peripheral membrane protein 22, all these involved in myelination and compaction of this myelin. So what is to be remembered is that in the PNS myelination, axonal myelination, there is centrifugal wrapping from centers towards the periphery. Okay, there is centrifugal wrapping of the Schwann cell mesaxon, so that the newest one will be towards the periphery and the oldest one will be towards the center. Now as I said, the Schwann cell cytoplasm is left during myelination in myelination process. So what are those pockets where the cytoplasm of Schwann cell is left? One is called the outer collar of Schwann cell cytoplasm, which contain the nuclei of Schwann cell, okay, nuclei of Schwann cell and some organelles. So this is called outer collar of Schwann cell cytoplasm, that lies just immediately external to the myelin sheath. There is one inner collar of Schwann cell cytoplasm, which is just located between the myelin sheath and the axon, and even inside the myelin sheath there are strands of Schwann cell cytoplasm which are called Smith-Lanterman cleft, and there is also cytoplasm at the node of Ranvier at the perinodal, which is called perinodal cytoplasm of Schwann cells. So this is one axon and its relationship to the myelin. So regarding node of Ranvier, what is to be known is that node of Ranvier is the site where two adjacent Schwann cell meet. This is one Schwann cell and this is another Schwann cell. Each Schwann cell myelinates just one segment of an axon, the length of which could be 0.1 millimeter. Okay, that is the segment of an axon that can be myelinated by one Schwann cell, individual Schwann cell, and that segment is called internode. Here the adjacent plasma membrane of the Schwann cell are not tightly associated, and so extracellular fluid can pass and have access to the axonal membrane, axonal plasma membrane, and these axonal plasma membranes are rich in voltage gated sodium channels. So this is the site of depolarization at the node of Ranvier. If we unroll this entire myelin sheath of this myelinated axon, this is one axon, we find that the inner collar of the Schwann cell cytoplasm, this is the inner collar of Schwann cell cytoplasm, is continuous with the outer collar of Schwann cell cytoplasm. This is the outer collar of Schwann cell cytoplasm through the Smith-Lantermann cleft. These are the Smith-Lantermann cleft and the perinodal cytoplasm. So this is the ultrastructure of a fully myelinated PNS axon. This is the myelin, outer collar of Schwann cell cytoplasm. This is lined with Schwann cell membrane, what we call sheath of Schwann or neural lemma, and external to this there is one external lamina of Schwann cell or basal lamina. So this is the external lamina or basal lamina formed by the Schwann cell. Now just outside the basal lamina you can see some collagen fibrils in cross sections. These collagen fibrils collectively form what we call the endoneurium, the lining of the individual axon. Endoneurium, the lining of the individual axon, we will come to this later on. Now while myelinated axons are nicely taken care of by Schwann cell, it is not the case with unmyelinated axons. The unmyelinated axons of the peripheral nervous system are also taken care of by the Schwann cells. Now here what happens, the Schwann cells form multiple groups onto their membranes, multiple groups into which axons come to lie. Now a single group can accommodate one axon or more than one axon, and if the Schwann cell is large enough, it can have more than 20 such pockets, each containing one or more unmyelinated axons for their structural support. So this is the pattern seen in peripheral nervous system, and in the autonomic nervous system, in the visceral system, it is common to find multiple axons, unmyelinated axons, say 20 to 30, to house inside each invaginated structures. Now in the CNS, the pattern of myelination is just different, because oligodendrocytes are the structures that are involved in myelination of the CNS axons. Now these cells are smaller than astrocytes and they have few processes. That is why they are called oligodendrocytes, and they are present in the white matter where they align to the fascicles, axonal fascicles. That is why they are called interfascicular oligodendrocytes. Unlike Schwann cells that myelinate just one segment of an axon called internode, here an oligodendrocyte can myelinate more than 30 axons at a time. So this is one oligodendrocyte. It has several tongue-like processes, each myelinating individual axons. So it can myelinate more than 20 to 30 axons with this tongue-like cytoplasmic processes. So here the myelination is just the reverse of what we have seen in the PNS. Here it is centripetal wrapping, that is, this wrapping starts from the periphery and then ends toward the center, encircling the axon. So that is also to be remembered. Now, not now, please. I feel disturbed. Okay?
Why, sir?
Okay, definitely I will tell. So in myelin sheath, you can see there are differences in the PNS and CNS. Like the Smith-Lanterman clefts are fewer in CNS because in CNS the myelin is supported by astrocytes, whereas these clefts are high in number in PNS. External lamina surrounding the axon is absent in CNS, whereas it is present in the PNS. The node of Ranvier is larger in the CNS, whereas this is smaller in the PNS because they don't fast conduct the impulses. They conduct impulses in a slower rate. Whereas ion channels are more towards the node of Ranvier, in unmyelinated axons, ion channels are present uniformly along their axons. So that is peculiar. So that is why nerve impulses is conducted rather slowly, unlike in myelinated axons where there is saltatory conduction because of the presence of channels at the node of Ranvier. And in unmyelinated axons, the impulse moves as a continuous wave of depolarization along the axons. If this myelin is disrupted due to certain reasons in diseases, like in autoimmune diseases called demyelinating diseases, there may be devastating diseases called multiple sclerosis in the CNS and Guillain-Barré syndrome in the PNS, both causing a slowing of axonal conduction. Now regarding central neuroglia, I have shown there are 4 types: astrocytes, oligodendrocytes, microglia and ependymal cells. In the CNS there is one terminology called glia limitans. Glia limitans is a composite structure formed by the astrocytic food process. These are the astrocytes. They are food processes here called sub-pial food process because this is the pia mater of the brain, pia mater that limits the brain. So this pia mater has a basement membrane. This is the basement membrane. This pink color is the basement membrane, and the subpial food processes. These two merge, forming what we call glia limitans. This glia limitans is an impervious structure and doesn't allow entry of molecules from the external environment. So this glia limitans is formed by astrocytic food processes. What are the other functions and the types of astrocytes? There are basically 2 types: fibrous astrocytes present in the white matter. They are less branched than the variety called protoplasmic astrocytes, which are much branched and present in the gray matter of the brain. Protoplasmic astrocytes. So they will encapsulate the neuronal processes, like here just over the myelin, called perineural end feet, and they will also terminate on the blood vessel wall called perivascular end feet. So these are the structures from the protoplasmic astrocytes. Fibrous astrocytes only form this kind of perivascular end feet. Okay. So among the various functions, they buffer potassium ions from the extracellular environment. They can uptake glutamate after neurotransmission is over. They supply lactate to the neurons, and these astrocytes also maintain blood-brain barrier by forming tight junctions between endothelial cells and synthesizing the proteins, tight junctional proteins by themselves. Now, when there is damage to the central nervous system, there is loss of neurons, and these neurons are then phagocytized, leaving an empty space. This empty space is then filled up by astrocytic processes. Astrocytes then proliferate, a process called astrogliosis. These astrocytic processes then form what we call glial scar that will fill the empty spaces. In the central nervous system, when there is excessive proliferation due to uncontrolled mechanism, so this leads to astrocytoma, a kind of neoplasm of the central nervous system, astrocytoma. Microglia are the smallest of this central neuroglia. Developmentally, they are called amoeboid. In the adults, they are ramified because they have several branches. And when they become activated in response to injury or need for phagocytosis, they become somewhat round with their processes withdrawn, and they contain lots of lysosomes in their cytoplasm. They are the brain's major immune cells, secreting cytokines, and also may function as antigen presenting cells. And lastly, the ependymal cells that line the brain ventricles and the spinal canal. So these are the ependymal cells made up of simple cuboidal to low columnar epithelial cells that line the fluid filled cavities of the CNS, brain and spinal cord. Their apical surface contains microvilli and cilia, structures that help in fluid transport, and apically they are interconnected by junctional complexes. Now these cells are specialized in the sense that these ependymal cells that line the brain ventricles and spinal canal, these epithelial cells lack the basal lamina. This is contrary to all other epithelial cells that rest on a basal lamina. And this basal part of the cells are highly interdigitated, and astrocytic processes penetrate into those interdigitations. So that is the unique feature of this ependymal cells, and this is just to transport materials rapidly. That basal lamina is excluded from this region. Now we will start about the practical aspect. First, we have to know how to see the neurons and their morphology. Now traditionally we use hematoxylin and eosin, to study the neurons, their shape and size, and also types if possible. So these are the Purkinje neurons. But here again you can see only the cell body is clearly seen, the processes are not clearly seen. This is the limitation of the staining method with hematoxylin and eosin. Now there are certain dyes collectively called Missel stain, like thionin, cresol violet and toluidine blue, that stain Missel substances or Missel bodies. So this will tell you the presence of Missel substances in the neurons, and this helps us to identify neurons, especially the large multipolar motor neurons that contain lots of these niche substances. There are other stains like phosphotungstic acid, hematoxylin, silver impregnation method as described by Camillo Golgi, and then osmium tetroxide is one agent that stains lipids, and so we can see nicely the myelinated axon. Okay. These are pictures from the cerebellum. Cerebellum has molecular layer, Purkinje cell layer, and granular cell layer, and internally there is a white matter.
Thank you.
What? You are not interested? So here the idea is to show you the Purkinje cells, because in the practicals you have about 7 to 8 slides that will tell you about the types of neurons, how the neurons look like. You have to identify the neuronal types, then the ganglia which are located outside the central nervous system, how do the ganglia look like, and the types of ganglia like sensory ganglia or autonomic ganglia of the autonomic nervous system, how the neurons are organized in the brain and spinal cord. In the brain, like in the cortex, in the cerebral cortex, in the cerebellar cortex, okay, and in the spinal cord. So these are to be identified in your practical classes. It is not difficult, but if you concentrate, I think it is easy to identify. Like here, when you see this kind of profile, molecular layer, Purkinje cell, and lots of granule cells, basophilic granule cells, you will see that in the Purkinje cell layer there are large neurons. These are multipolar neurons having extensive dendritic branches that penetrate the molecular layer. So once you identify this kind of neurons, multipolar neurons, you can tell that this is, or this slide is from the cerebellar cortex that you have to write. Now this is the same area, I mean the cerebellar cortex, but stained with phosphotungstic acid hematoxylin. Phosphotungstic acid hematoxylin additionally stains the fibrillar materials, which ordinary hematoxylin cannot stain. So here you can see this is a low power view and high power view, that this phosphotungstic acid hematoxylin has stained many dendritic processes and also other fibers that are climbing towards the molecular layer of the cerebellar cortex. This is the granular cell layer, contains granule cells. Now sensory neurons and motor neurons are present in the sensory cortex and motor cortex, and there are two slides where you can see them and you can identify them. Sensory neurons are located in the primary sensory cortex and motor neurons in the primary motor cortex. So this is from sensory cortex, primary sensory cortex, slide stained with hematoxylin and eosin. Here you can see the predominant cell type is granule cells, and these are all sensory neurons. So these are the granule cells of the sensory cortex. Okay. On the other hand, if you see the motor cortex, this is the histological organization of motor cortex as drawn first by Cajal, a Spanish neurocytologist. Here you can see there are 6 layers: molecular layer, external and internal granular layer, external and internal pyramidal layer, and polymorphic cell layer. Here you can see multipolar motor neurons like the pyramidal neurons. So these are examples of motor neurons that can be seen in motor cortex. The large motor neurons, multipolar motor neurons, are pyramidal neurons which are present in the layer 5th of this internal pyramidal layer of motor cortex. So this is one low power view of the motor cortex where you can see the multipolar motor neuron. Although this is not nicely stained, but you can still appreciate that it has a type which is not round and has many poles, like processes emerge from this part and also from this part, and also processes emerge from this part. So these are the apical dendrites of the pyramidal neurons. This is the cell body and apical dendrite of the pyramidal neuron. So when you see this kind of profiles in hematoxylin eosin preparations, you can identify them to be multipolar motor neurons, and if you can identify the layers, definitely this will be from the motor cortex. So there is no confusion. The small cells are neuroglial cells, central neuroglial cells. Then there is one method by which you can stain the entire profiles of neurons, like these are all multipolar neurons stained by silver impregnation method. So this is Golgi staining of the multipolar neurons, motor neurons. And this is one profile of multipolar neuron, pyramidal neuron, stained for one particular antigen present in the microtubules, dendritic microtubules. These are the dendrites, apical dendrites and basal dendrites. So dendrites contain lots of microtubules, and these microtubules are composed of microtubule associated protein 2. So when we apply antibody against this MAP2, the dendritic branches can be seen nicely. Then you can argue why the cell body is stained, because the cell body also contain lots of microtubules. So that is why the cell body is also stained for MAP2. So this is one method called immunohistochemistry. And here we have used fluorescent dye, so that is why it is called immunofluorescence method of antigen detection in brain sections. So this is one pyramidal neuron stained for MAP2. Here you can see that these dendritic branches are extensive, and if you see in high magnification, you will find that each dendritic process gives up several short branchlets called spines, stubby spines. So these spines are actually telling you the activity that is happening within the dendritic processes. Means more activity and more spines. When you learn, when you attempt to learn, and when the dendrites process information, there will be lots of spines on the dendritic shafts, dendritic spines. But as you start forgetting, these dendritic spines will regress. So a dendrite can have more than 5,000 to 6,000 spines, such kind of spines. So more spines, more activity. Then one type of multipolar neuron can be seen in the spinal cord. In the spinal cord, you may be familiar with that, whereas in the brain, the grey matter is external and white matter is internal. The situation is just reverse in the spinal cord. In the spinal cord, the grey matter is internal. This is the grey matter in the form of a butterfly in the spinal cord. The grey matter is internal and the white matter is external. This grey matter is divided into three compartments: dorsal horn, sensory, lateral horn, motor, and ventral horn, motor. Okay, means containing motor neurons, multipolar motor neurons. So these two parts are interlinked by grey chromosphere. Here you can see, and you might know that it is incompletely divided into two halves. Spinal cord is incompletely divided into two halves by a ventral median fissure. Ventral median fissure where the pia invaginates, where the pia and blood vessels invaginates. Okay. So this ventral median fissure and its presence indicates the ventral aspect of the spinal cord. Means sometimes you may confuse which one is dorsal and which one is ventral. But this ventral part is broader and it has a fissure called ventral median fissure. So this is one low part view of spinal cord showing dorsal horn, ventral horn and the white matter. This white matter is the region containing myelinated and unmyelinated tracts. These tracts reach to the brain, and also tracts that leave the brain and come to lie in the spinal cord also are located in this region of white matter. Now our target is the ventral horn cells or anterior horn cells in the ventral horn of the grey matter. This is the central canal of spinal cord lined with ependymal cells, and scattered among these are the neuroglial cells. You can see neuroglial cells, central neuroglia. And now we are showing the ventral horn grey matter of the spinal cord that contains large multipolar motor neurons. So large multipolar motor neurons, this can be seen nicely even when we prepare with some Nissl stain. Okay, so these are the multipolar motor neurons, and there are lots of Nissl substances. And here you need to see what we call one kind of meshwork. This meshwork is called neuropy. This meshwork is formed by the processes from dendrites and axons and the central neuroglia. So this neuropil is also nicely stained with the Nissl dye, Nissl stain, like thionin or crystal violet. Peripheral nervous system. In the peripheral nervous system, you have slides that show the organization of peripheral nerves. This peripheral nervous system comprises the peripheral nerves with the specialized nerve endings, the receptors, and the ganglia that containing the nerve cell bodies. This ganglia is located outside the CNS. Now, what is a peripheral nerve? A peripheral nerve is a collection of nerve fibers, okay, is a collection of nerve fibers that are held together by connective tissue sheaths. So, what is the organization of peripheral nerve? There are individual axons that cluster together forming an axon fascicle. And there can be multiple axon fascicles in a large peripheral nerve. This is one axon fascicle, this is another axon fascicle, and all these axon fascicles are then combined together with a connective tissue sheath forming the peripheral nerve. Now, at every level, the individual nerve fibers are supported by connective tissue sheath. That we must know at this stage. So, what are the various connective tissue components of a peripheral nerve? The individual nerve fibers are held together by connective tissue that are organized distinctly into three compartments called endoneurium, perineurium, and epineurium. Endoneurium is the connective tissue sheath that surrounds the individual nerve fiber, endoneurium. Perineurium is the thick connective tissue sheath that surrounds individual nerve fiber fascicles. So there are multiple axons that are held together by this perineurium, and the whole nerve is then covered by what we call epineurium, which is a thick connective tissue sheath, a type of dense irregular connective tissue sheath that lines the entire nerve and also penetrates the substances of the perineurium. Here you can see the epineurium traverses up to the level of perineurium. Now, this endoneurium is made up of collagen fibrils, and since fibroblasts are rare in the endoneurium, it is most likely that the collagen fibrils are secreted by the Schwann cells that lie in association with the nerves. Perineurium, on the other hand, is somewhat specialized. Perineurium has multiple layers, and these cells look like epithelioid, means they have external lamina, and the cells are held together by tight junctions, and their cells contain lots of actin filaments, that is, they look like somewhat smooth muscle cells. Again, in the perineurium, like this is the one, perineurium that is bordering this axon fascicle. In the perineurium, no fibroblasts are seen. So it is likely that this perineurial cell actually synthesize and release collagen molecules in the basement membrane. And finally, the epineurium, it is a type of dense irregular connective tissue, and penetrates the interstices of this perineural sheath, and blood vessels are present in the epineurium and that travels up to the level of the perineurium. At the level of endoneurium, blood vessel is very less, and because of this poor vascularization, the nutrients and important metabolic substances are transported from the epineurium to endoneurium across this perineural border. So endo, peri, epineurium can be identified in a peripheral nerve, and this is one method. If we fix the peripheral nerves in osmium tetroxide and stain with toluidine blue, then we can identify the myelinated axons nicely, and node of Ranvier and Smith Lanterman cleft nicely. Okay, so these are the myelinated axons cut in cross section. The diameter is variable, indicating that there are several types of axons, and these fascicles are lined externally by epineurium. This is the picture of longitudinal section of this myelinated axons. Here you can see the myelin sheath, and where this myelin sheath is broken, that region is called node of Ranvier. And you can see also Smith Lanterman cleft, which are the strands of Schwann cell cytoplasm. There are also slides where you can see the peripheral nerves are cut in longitudinal section in this way. So if they are cut longitudinal section, you can see the perineurium clearly as a dense band of connective tissue, and this is the axon fascicle containing the nerve fibers, and you can also see the wavy patterns of these axons. Wavy patterns of these axons, and in between you can see the Schwann cells along with these axons. So this is one axon fascicle and this is another, and these individual fascicles are lined by perineurium. So you can identify that this slide is from a peripheral nerve. Somewhat similar is the picture like this. This is the axon fascicle and lined by perineurial sheath, and these are the Schwann cells. So this is another picture where the axons are cut in cross section, and here the axons are imperfectly cut in longitudinal section. This is in fact a drawing. So this is one axon fascicle. It is lined by perineurium, perineurial sheath, and the cell of the perineurium. It should not be confused with Schwann cell, because Schwann cells never lie at the level of perineurium. This could be one Schwann cell, and these are the individual profiles of myelinated axons. The axons lie in the center. This is the axon, and the whole thing is the myelin sheath, but this myelin is dissolved out during tissue preparation, leaving what we call certain spike-like structures called neurokeratin. So these are nothing to do with keratin molecules of the skin epidermal cells. They are simply supportive structures of these neurons. And externally you can see one lining that is called neurilemma or sheath of Schwann, containing the Schwann cell nucleus. And same is the case when you see in longitudinal section. This is one profile of axon, myelinated axon. This is the endoneurium, and this is the Schwann cell nucleus. This is the perineurium border. Now regarding this could be one fibroblast cells, because Schwann cells do not lie in the endoneurium. So I think you can identify the peripheral nervous system neurons and their fibers, the peripheral nerves, be there in cross section or longitudinal sections, by seeing these features carefully. Now these are two pictures of peripheral nerves stained with osmium tetroxide. Okay, osmium tetroxide. As I told you, osmium tetroxide stains myelin nicely, and you can see myelinated axons clearly. So these are the axon fascicles. These are the individual axon fascicles. These are the individual axon fascicles lined with perineurium. So this is the perineurial sheath, and when we see one section cut in longitudinal profile, so here you can see this is the axon fascicle containing myelinated axons. These are the myelinated axons, and this fascicle is lined with perineurial sheath. So this is the perineurial sheath, so clearly identifiable. Then there are slides where you can see the neuronal types in sensory ganglia and autonomic ganglia. Sensory ganglia, as you know, carry sensory information toward the CNS, and autonomic ganglia or sympathetic and parasympathetic ganglia carry motor information to their effector cells. So this is one low power view of dorsal root ganglia, section from dorsal root ganglia, where you can see the neuronal cell bodies are clustered together. Okay, they are clustered together. So these are the sensory neurons and unipolar neurons. Now if you see carefully, you can see there are certain features by which you can identify the sensory neurons in a sensory ganglion like dorsal root ganglion. So each of these sensory neuron has a large cell body with a distinct nucleus that is centrally placed and a prominent nucleolus. So note here, the nucleus is exactly in the center of the cell body of the sensory neuron, and the entire cell body is nicely invested by the satellite cells. These are the peripheral neuroglia. These small dots are these peripheral neuroglia as satellite cells that invest the entire cell body of this sensory neuron. Okay, here in the sensory ganglia there are no synapses, at least so that the peripheral neuroglia can intimately contact the neuronal cell body for their metabolic support. So they are intimately associated with the cell body of these sensory neurons. This is the diagrammatic view of the sensory neurons in a dorsal root ganglion. This is the centrally placed nucleus with prominent nucleolus. These are the satellite cells, and the other nuclear profiles are Schwann cells that lie in association with the fibers, because fibers here come and they also leave to reach to the dorsal horn of the spinal cord. So these are the fiber bundles. As these neurons are clustered together because each neurons may have a different characteristics, some carrying pain sensation, some touch or temperature like that. So accordingly the size and their neurotransmitter characteristics differ. So these are the features of sensory neurons in dorsal root ganglia, and this is one from your autonomic ganglion. Here this is one sympathetic ganglion which contains motor neurons, and these motor neurons are multipolar neurons. In the sensory ganglia where sensory neurons are located, these sensory neurons are unipolar, but here in the autonomic ganglia or sympathetic ganglia, the neurons which are motor are multipolar. They are not unipolar, they are all multipolar. Now little bit about this autonomic ganglia you must know at this stage. Detail you will read when you read the nervous system in detail, that we know about the somatic efferents. Somatic efferents means the motor fibers that convey impulses via the ventral horn of the spinal cord to their effector cells like skeletal muscles. Here only one neuron conveys information from the spinal cord to the effector cell, skeletal muscle, for muscle contraction. So there are no intervening neurons here. Whereas in case of autonomic system with visceral efferent neurons, these neurons which originate in the lateral horn of the spinal cord, see this is the lateral horn of the spinal cord, the fibers called presynaptic or preganglionic fiber first come to lie in a ganglion called autonomic ganglion or sympathetic ganglion. Here this is one paravertebral ganglion lying close to the thoracic level of the spinal cord. This paravertebral ganglion will make synapses with this presynaptic fiber, preganglionic fiber. Lots of synapses will take place here within this ganglion, and from this ganglion the fibers will emerge. These fibers are called postganglionic fibers or postsynaptic fibers that will ultimately innervate their target effector cells like the smooth muscle cells in blood vessels and the glandular epithelium, for example in the sweat glands. So we are now at this level of a sympathetic ganglion carrying motor information, and as you can understand from here, these are all postsynaptic neurons because presynaptic neurons are located here and the information goes postsynaptically from this ganglia to the effector cells. So these are all postsynaptic neurons and they are multipolar because you can see multiple projections, although not clear, but they are not uniformly spherical. Preparation is not good, but you can still identify the multiple branches of these multipolar neurons, and these are all motor neurons, and they can be discriminated from unipolar neurons by the location of their nucleus. The nucleus is not right in the center. Unlike in the unipolar neurons where the nucleus is located right in the center of the cell body, here the nucleus is located towards the periphery. Technically that is called nucleus is eccentric, like here you can see one, another one, and another one, nucleus is pushed towards the periphery of this cell body of this multipolar neurons. And here, since these neurons receive synapses from the presynaptic elements, so these satellite cells are not intimately associated with the neuronal cell body, a feature that is characteristically different from the unipolar neurons that receive intimate association of these satellite cells. Okay, so these are the features of the sympathetic ganglia with multipolar neurons. And with this you can see there are some associated fibers that contain the nuclei of Schwann cells, and here this is the connective tissue sheath, connective tissue capsule of this ganglia. This is low power view, high power view. There are certain profiles of vein, arterial present in this ganglion. So with this we come to this end of this lecture, and if you have questions you can now ask. Any questions? Louder, please.
Sir, what do you mean by fast anterograde transport? SCR, vesicles and mitochondria get transported from cell body to axon.
Right.
Is it something like that?
Yes, fast anterograde transport, by which these membrane bound structures like smooth endoplasmic reticulum, mitochondria and synaptic vesicles are distributed to the axon terminals. This is anterograde,
Sir, and what?
fast anterograde transport.
Kinesin and,
Pardon?
kinesin and dynein, what are their functions?
These actually microtubule associated proteins. They bind with microtubules, and microtubules need some energy for this transport. So these are all motor proteins that at the expense of ATP carry out this journey and transporting the materials from one place to the other, from cell body to the axon terminals and also back to the cell body. So in the anterograde, kinesin is involved, and in the retrograde transport, dynein molecules are involved.
Okay, sir, I got it. Thank you so
Yes,
much. Sir.
yes. See, there is no scope to explain everything in detail because of time. But if you see Ross, you can find there is a picture where you will see how microtubules bind to this kinesin or dynein, and how the transported molecules then bind to this microtubule and microtubule protein complexes for their transport. You can see that.
Fine, sir. I see. Thank you, sir.
Okay, thank you.
Excuse me, sir.
Yes,
Sir, this anterograde and retrograde transport is also applicable for
Uh, yeah, should be, but somehow this is not described in the literature. But what is described is that the first retrograde transport means from axon terminal to cell body and from dendrites to cell body. Nothing extra is not mentioned in any books so far. Although transport goes to all directions, but since this can be nicely seen even in live neuronal culture, where you can see the axonal transport going on in vivo, time lapse, video, microscope, you can see that this is more prominent in the axons. Maybe that is why this term axonal transport has been coined. But the entire process is transport of materials to all parts of the neurons.
Okay, sir.
Only thing that you need to remember is that this slow axonal transport is always anterograde. It has no bidirectional movement. So that is important also from exam theory point of view.
Okay, sir.
Thank you.
Thank you.
And the reason for this slow axonal transport is always anterograde.
No, that is just a physiological division of labor so that the molecules like actin monomers, tubulin monomers, and monomers for these intermediate filaments, that is neurofilaments, which are present in the axons. So these monomers, which are the backbones of this cytoskeleton, these proteins are, or their monomers are transferred very slowly. It means it is time dependent. They cannot be transported fast. So they are transported slowly towards the axonal end. So that is why slow. And in fast axonal transport, these organelles and can be transported efficiently with a faster speed, and they also need a faster speed for faster neurotransmission to occur. So smooth endoplasmic reticulum should be there at the axon terminals, and synovial vesicles are to be there, and mitochondria are to be there for energy liberation. And once neurotransmission happens, then the synaptic vesicle membranes are retrieved via that clathrin dependent endocytosis, and then these vesicular membranes are packaged in the form of synaptic vesicles, and these synaptic vesicles may package new synaptic materials right at the axon terminals, or these vesicular membranes may be transported towards the cell body, where new synthesized neurotransmitter materials can be repackaged at the level of cell body, and again they will be transported anterogradely to the axon terminals. This is how the system works.
Sir, and what are ribbon synapses? Can you explain it again?
What are
Ribbon synapses.
ribbon synapses?
Yes, sir.
Ribbon synapses are present in the photoreceptor cells of retina, in the pineal gland, and in the hair cells of cochlea. So these are some folded plasma membrane that appears during differentiation of photoreceptor presynaptic terminal, and they are specialized, means they are specialized in such a way that they have some proteins, and these proteins always attract the synaptic vesicles, and the synaptic vesicles then bind to these ribbons, and then they allow these vesicles to dock and fuse to the active zones of the presynaptic membrane. So these ribbons actually behave like what you see in the airport, where the conveyor belt carries our luggage materials one by one. So in that way, these ribbons have been designed by nature in the photoreceptors, means so that there is a very slow regulated release of synaptic materials to the postsynaptic terminals, because you know the photoreceptors and hair cells, they work continuously, so neurotransmitter release should be in a very controlled fashion, so that the system is not depleted of neurotransmitters. That is why nature has devised this kind of slow transport mechanism, by which these vesicles will come slowly and fuse with these ribbons, and then they will dock and fuse with the presynaptic membrane, and then exocytosis will happen. So this is the characteristics with the ribbon synapses seen in some regions of the central nervous system.
Thank you, sir.
Okay. You can also read little bit from the photoreceptors, like I must say at this stage, because I am specialized in this field, that whereas a depolarization is a must for an action potential to propagate along the axon, in case of photoreceptors, depolarization is not needed. It is hyperpolarization that actually triggers neurotransmitter release via exocytosis. So this is unique. This is again the interplay of nature, that hyperpolarization has been induced in photoreceptor cells for neurotransmission, because with hyperpolarization the wave of electrical impulse is slow, but still it is just good enough to allow very slow release of neurotransmitters across the synaptic cleft, a phenomenon which is called quantal release of neurotransmitters, very small limited amount of neurotransmitters, which is just good enough to carry out the impulses from presynaptic to the postsynaptic units. So these photoreceptor cells work via generation of hyperpolarization, unlike other neurons that act via depolarization. And the Japanese scientist who first discovered this mechanism, that light hyperpolarizes photoreceptor cells, in 1951, Edward Tomita got Nobel Prize for this excellent discovery, that hyperpolarization is the basis for photoreceptor neurotransmission. So this is exceptional. So at this stage you must read, and gradually you will read all this information on the characteristics of different neuronal cell types, their features and the physiological properties of these neurons, how they behave and act in different systems.
Acad section has removed those people from the thing.
Who?
Dean Acad section.
हाँ.
जैसे वो मैसेजेस आ रहे थे, हमने स्क्रीनशॉट लेके मैंने जाके शरीफ सर को दिखाया.
हाँ.
फिर सर ने बोला गो एंड कंप्लेन इमीडिएटली टू द डीन एकेडमिक,
हाँ.
राजीव कुमार सर.
हाँ.
तो आई शोड हिम.
हाँ.
उससे पहले एक और भी मेल था जिसमें ये सब हो रहा था.
उल्टा पुल्टा. हाँ.
हाँ हाँ, तो वो सब स्क्रीनशॉट्स थे उनके पास. तो उन्होंने तुरंत सबको मैसेज कर दिया कि कल से नेक्स्ट क्लास से 15 मिनट ज्वाइन होने के बाद द मीटिंग विल बी लॉक्ड. दिस विल बी ऑन रिकॉर्ड.
नहीं नहीं, मैं वो करके रखता.
यूट्यूब में रिकॉर्ड हो रहा है.
अच्छा, तो बंद कर दो.